Okay, good morning, guys. I'm going to make a quick video on mitosis and meiosis. Somebody requested information about it, so I'm just going to kind of go over it again so that you have it in another form from me now, rather than just reading it or watching a video um, from someone else. Now you're getting it in a third way. Okay. Um, they're very similar, but yet they're different. Okay, so you're still going to have the same stages that you're going to go through. Mitosis is going to be one division, and meiosis is going to be two divisions. Okay, um, the names of the stages are the same, and they're going to be essentially the exact same thing happening in each stage. Okay, so it's not like prophase and mitosis is totally different from prophase and meiosis. So just keep that in mind. Okay, so let's go over mitosis first because it's the most basic, it's the easiest one to grab onto. So mitosis happens in every type of cell. So your skin cells, your liver cells, your heart cells, all of those cells are going to go through mitosis. That's the only way you can make new cells. So every time you scrape off dead skin cells from your skin cells are going through mitosis all of the time, replacing all of those dead skin cells that you're losing. Okay, so that's what's happening with mitosis. That's where this is coming into play. Okay, so it's going to go through these phases, and at the end of it, you're going to get two identical daughter cells that are going to look exactly like your original cell that you started with. Okay, that's the big thing about mitosis is you get two identical daughter cells. Okay, so we're splitting that nuclei and we're getting two identical at the end. Okay, in prophase here, okay, the spindle fibers are starting to show up, okay, at the edge, and then your DNA is going to start to condense. Your chromosomes are condensing. Okay, we've already replicated the DNA because that happens in interphase. Okay, so that's already done. Now it's condensing and it's coming together. And then we're going to move into metaphase, where our chromosomes are going to line up at the very center. Meta, think of middle. That's the easiest way. Okay, they're going to line up straight down the equator of the cell. Okay, and these spindle fibers are going to kind of place them there, so they're connected at that same time. Okay, the next phase then is anaphase. This is where they're going to pull apart. So we have our chromatids are separating in that chromosome and they're pulling apart. Now remember, each of these chromatids contains identical information. So whatever is on this one right here is going to be on this one here. Okay, so they have identical genetic information. And they're going to move to separate sides so that now we are getting those two cells that have the exact same information. Okay, and then in telophase, okay, all of our genetic information has moved to the sides. It's totally over towards each pole. Okay, those spindle fibers start to disappear, and then that nuclear envelope starts to come back in. So this is like telophase and cytokinesis in one right there. Okay, I just put them together rather than drawing a whole separate one. And then what we get at the end are these two identical daughter cells. And then those ones would go on to split as well. Okay, those would go on to split and create the same exact type of cell. Okay, so it's just a never-ending cycle. Okay, however, there are things that control the cell cycle to make it stop. Okay, so because if you think of a tumor is uncontrolled cell growth. So what's happening is those cells are going through mitosis over and 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 never stopping. Whereas, other types of cells, all types of cells go through the cell cycle and they know when they need to stop and when they need to make new cells. So a tumor is just a cell cycle that has gone awry and is not following what it should be doing. Okay, so that's mitosis. Okay, I hope that helps in terms of that. Let's go on to meiosis next. Okay, all right, here we go, meiosis. So there's still interphase that occurs before meiosis, just like in mitosis, okay? So DNA is being replicated, proteins are being produced, and so forth. That's happening in interphase. Now, when we talk about meiosis, you got to keep in mind the difference between homologous chromosomes and sister chromatids. Okay, so when we're in prophase 1, we're going to have homologous chromosomes. Okay, so we are replicating that DNA from, the mother's chromosome, and then we're also replicating the DNA from the father's chromosome. So we're going to have 2 copies of those. Okay, and that's what we're going to end up here with in prophase 1. So when we're talking about meiosis 1, whoops, I spelled it wrong. We want to talk about homologous chromosomes. A homologous chromosome is made up of sister chromatids. So I'm going to draw it here for you. These 2 together are homologous chromosomes. Okay. So this would be like the mom and the dad. Then each of these are called a sister chromatid to each other. Okay, so mom is made up of 2 sister chromatids, and dad's chromosome is made up of 2 sister chromatids. Okay, so I hope that clears that up. Make sure you understand the difference of those, okay? So prophase 1, our chromosomes are condensing, so we're starting to see them become visible. And then, I didn't draw this in, our spindles are starting to be produced, okay, at the centrioles, okay? So we've got that going on. Now, metaphase 1 is just like metaphase of mitosis in terms of where they're lining up. Metaphase 1 just means that those homologous chromosomes are going to be sent to the center by the spindle fibers, and they're going to line right up, okay? So you see I have 2 chromosomes, 2 homologous chromosomes lined up in the center, okay? Anaphase 1, now those homologous chromosomes are going to separate and go to the sides. Okay? Now, in metaphase 1, okay, this is where crossing over is going to occur. Okay? Crossing over will occur here, and so will random assortment. And I'm going to show you a picture from your book about random assortment, but I'll talk about that in a minute, okay? So crossing over, all that's going to happen is literally like the leg of a homologous chromosome is going to like flip-flop. That's all that happens. Okay, so nothing major, but it is just going to like flip-flop a little bit of genetic information where, now when I say that, I mean that it's still giving the same gene, but maybe instead of on mom's chromosome, now it's going to have a portion of dad's, and I'll show you a picture of that too. So let's stick to meiosis 1. Sorry, getting off on a tangent here. So metaphase 1, meta, remember, is the middle. So they're lining up. Those homologous chromosomes are lining up in the middle. We go to anaphase 1. Now the homologous chromosomes are separating. Okay, they're going towards the poles of each of the sides of the cell. And then in telophase 1, now we are on our sides of the cell, and we're going to, we're in telophase 1, so that nuclear envelope is coming back around the genetic material on both sides of the cell, and then cytokinesis is going to cleave, and we're going to have 2 cells, okay? There's a brief interphase in between meiosis 1 and meiosis 2, but there is no DNA replication that happens. Only some creation of new proteins is going to happen in there, okay? So then we go to prophase 2, okay? DNA is going, or the chromosomes are going to condense again and become visible, okay? That nuclear envelope disappears, okay? We go to metaphase 2, meta again. We're lining up in the middle, okay? So you can see that our chromosomes have lined up in the middle. All right, then we go to anaphase 2. This, remember, meiosis 2 is exactly like mitosis, okay? Anaphase 2, now those sister chromatids have pulled apart, okay? So exact copies are moving to each side. Granted, they'll have a little bit of genetic variation from the recombination or the random assortment and the crossing over. Okay, so anaphase 2, they're moving towards each pole. And then telophase 2, the nuclear membrane is coming back around that genetic information in each side of the cell. And then we're going to have cytokinesis that's going to come in and cleave those into 4 cells. So now we get, just do that, 4 genetically different gametes, so 4 eggs or 4 sperm, okay, and so totally different from what's going to happen in mitosis, where we get 2 identical daughter cells, okay? I hope that helps. The next video coming up is going to be crossing over and random assortment. Okay guys, so let's look at crossing over. So crossing over is going to occur in prophase 1. I said in the last video metaphase 1, so I apologize. Prophase 1. Okay, so this is where those chromosomes, everything's condensing and becoming visible, and so they're coming together and they're going to start to kind of cross over. Okay, so here you can see we have these homologous pairs. Okay, here are your sister chromatids that are identical. It'll be easier to show you here. Okay, they have the same exact genetic information. Okay, and here's the homologous pair. Same genetic information and on each of these here. Okay, but see how they have A and A? So it's the same gene, it's just from mom and from dad. Okay, so then what will happen is crossing over can occur here. Okay, so all they're doing is switching genetic information. So portions of these homologous chromosomes are just going to swap places, and it creates genetic diversity because now we're just giving a unique combination of alleles. So as we go through meiosis 1, these separate, right? Okay, here's our example. Oh, excuse me. So now we have our chromosomes separated, so they're going to go through meiosis 2. Now these sister chromatids are going to pull apart, okay, and then we end up with an all blue here, and instead of another all blue that's exactly the same as this, now we have a blue and part of a pink. So they're not identical anymore. Okay, we've created some genetic diversity. Same here. We've created some genetic diversity. So now we have 4 different gametes, essentially, just with that one chromosome. So think about if all 23 23 pairs did that in us. Okay, then we would have an enormous amount of genetic diversity if all of them were crossing over. Okay, now let's see if it will let me turn pages. Now let's look at random assortment. Okay, so this is going to happen in metaphase 1, which if you look, you see them lined up in the center. Okay, so that's metaphase 1. And all that's going to happen when it says random assortment is they're literally randomly assorting themselves along the center of the cell. So with these 3 homologous chromosomes, we're going to see that there's 4 alternatives. So in alternative 1, we're going to have all blue and all pink on one side. So they randomly assorted themselves this way. Let's say that. Alternative 2, now we have homologous chromosomes 1 and 2 are the same place as they were here, but in chromosome 3, homologous set here, the blue flipped and is just lining up on the bottom part now. Totally random, just the way it assorted itself. And then you can see the difference, okay? For alternative 1, here's what one cell looks like and the second cell looks like. Whereas in alternative 2, here's what your first one looks like. See how it's different? And your second one. See how it's different. Okay, we've just created genetic diversity by them randomly, totally different lineups, okay? Alternative 3, now we're going to have a blue 1 on top, blue 2 on bottom, blue 3 on top. And same thing, we're going to get different gametes. In alternative 4, now only blue 1 is on top. Okay, blue 2 and blue 3 are now on the bottom, and now we get 2 more totally different gametes. Does that make sense? I hope that makes sense. Okay, so all it is doing is they're randomly, I mean, just think of the two words, randomly putting themselves in the center of the cell. Okay, homologous chromosomes are still going to line up next to each other. It's just whether or not it's going to be on this side or they're going to flip flop and go this way. Okay, and that happens differently every single time um, the cells go through meiosis. They can randomly line up different every single time. So every month when a woman is dropping an egg, they're going this meiosis process has happened, and so now all of those gametes are different. Okay, so that doesn't even include once they've lined up and now they have they have crossed over in prophase 1. So think of every time they cross over in prophase 1 and then randomly line up, all of those differences that are possible. Okay, that's how we get such genetic diversity, even among siblings. Think of you and your siblings. Maybe you don't look exactly alike. Okay, that's how we get that. So know that crossing over is going to happen in prophase 1. So all they're doing is like swapping a little leg. And then random assortment is going to happen in metaphase 1. Okay? And it's just where they're going to line up. Are they going to line up top to bottom or next to each other? Or is it going to be flip-flop this way and then this way? Okay? If you have questions about that, just make sure you ask.